go, guys.
What's up? You?
Hi, my name is Nick Lubin, and I am giving an informative speech on embryonic stem cell research. So when people hear the phrase stem cell research, negative connotations come to mind, like human cloning and embryo dissection. But there's a lot more to stem cell research than that. Stem cells can be obtained from embryos and other things, but I'm going to focus on embryonic stem cells because embryonic stem cells are the most controversial form, they're the most talked about in the field, and they are also the most useful for medicine research. I'm going to be focusing on how these embryonic stem cells are obtained, what they are, how they can be used for medicine, and how they are used for research. So embryonic stem cells, like the name would suggest, are obtained from embryos, and these embryos are obtained from a few different sources. One source is through in vitro fertilization. There can be leftover frozen embryos that can be donated for embryonic stem cell research. Another source is somatic cell nuclear transfer. This is where eggs are collected and then a person's nucleus is taken from one of their cells and planted inside that egg to create the embryo. The last source that can be used is existing stem cell lines. These existing stem cell lines are formed from embryos, and the stem cells from this embryo is proliferated to create millions of more stem cells that can be used and replaced as needed and can continue as a lasting line indefinitely. So, stem cells found in embryos are the most basic kind of cell with incredible potential. At stemcells.nih.gov, they explain that stem cells have three unique characteristics. They can proliferate for long periods, they are unspecialized, and they can be induced to become specialized cell types. Proliferation happens when cells continually divide and proliferate into more unspecialized stem cells for a long period of time, producing millions of stem cells from an original small number of them. What it means for stem cells to be unspecialized is that they are not any kind of cell found throughout the body that functions to create a certain tissue that is used in a certain way in the body. They're not cells that make up muscle, nerve, stomach, or liver tissue. It's a blank slate that can become any kind of cell in the body. They are also useful for research in medicine because they have not realized their final property, their ability to be induced into becoming specialized cell types. The embryo uses the stem cells to generate every type of cell in the human body. Beyond just being unspecialized cells capable of becoming specialized cells, embryonic stem cells are unique from most other types of stem cells because they are pluripotent, which means they can become any type of cell found in the human body. Many stem cells are only multipotent, meaning they can only become certain types of cells in the human body, and this is what makes embryonic stem cells much more valuable for medicine research. Embryonic stem cells are valuable for a type of medicine called regenerative medicine. Johnson, in his book Bioethics Beyond the Headlines, explains that embryonic stem cells are capable of replacing dead cells, forming new cardiac muscle, healing spinal cord injuries, and even healing degenerative diseases like diabetes by replacing the islet cells that aren't functioning properly. Stem cells have been successfully applied to several spinal cord injuries in mice, reconnecting the nerves, and they're able to walk again even after the spinal cord has been severed because of this healing process. James C. Peterson, in his article, Is a Human Being is an Human Embryo a Human Being, talks about the potential for producing whole organs that can be transplanted into patients that are exactly identical in DNA to the person, and therefore there will be no immune rejection. So there's a lot of potential for regenerative medicine and organ transplant with embryonic stem cells. Beyond regenerative medicine, which might not be realized until long into the future, there's so much to be learned from studying embryonic stem cells. We can learn about gene activation, how stem cells program to become specific cells and to form specialized tissues of the human body. And another area that can be understood through embryonic stem cells is early development. Knowledge might be gained regarding fertilization and fetal development, genetic defects and their origins, and the processes of diseases. Grasping the origin of genetic defects and discovering ways to prevent this formation could improve the quality of life for many humans who would otherwise suffer from the mental and physical disabilities caused by genetic defects. Research done on diseases is an important area of research as well. For instance, the process of cancer formation is a central one to study with stem cells. If someone could discover the source of many cancers, learn the process of their formation, and ways to prevent it from happening, many lives could be saved. And this is a potential that embryonic stem cell research has. Overall, embryonic stem cells are very unique cells collected from embryos, and they have amazing potential in regenerative medicine, and they can be very useful for medical research in the future. Embryonic stem cells have opened up doors in cellular biology and regenerative medicine that can make huge changes in medicine as a whole in the future. As long as research is conducted responsibly, this is a bright future for embryonic stem cells. Thank you.